Perfect. Okay, so thanks all for coming. It's my great pleasure to welcome Thomas Christophel today. Thomas Christophel.
I don't know how to pronounce myself.
He is currently an Hermeneuter group leader at the Bernstein Center and at the Department of Psychology, using the scanner at the UCAN, the Berlin Center of Advanced Neuroimaging Studies also. And Thomas has been in Berlin for a very long time. I think in 2008 he completed his diploma in psychology in Bremen, and then you kind of moved here, did a dissertation here with John Dylan Haynes. Is that not correct? Yes, that's correct. You look skeptical. And in 2013 you finished that, and after that you were a postdoc also here at the Charité and at the Bernstein Center for Computational Neuroscience until last year when you became a junior research group leader. That's you now. So welcome. There's a window that popped open. I don't know if that's a thing.
Yeah, that's just it being recorded.
Okay. Good. So I think you can take it away. I'm very happy to have you here. The talk today is going to be about working memory, which is the topic that Thomas has been working about. If you have any questions about fMRI research or methodology, he's also your guy. Yeah, famous, world famous machine learning stuff, and yeah, go ahead.
Yes, welcome. This is a talk about magic. Also, I'm like in Martin's world, I'm the methods guy. In my world, there are other methods guy, and I'm the psychologist. So, right, there is that beautiful gradedness of expertise that we have. I actually realized that if I stand in the camera, you're not going to see the screen, so I'm just going to stand here, and my camera is just going to be there. Yeah, welcome. This is quite exciting to be here. Before I start talking, I'm going to acknowledge the fact that this is the first time in 2 years holding such a thing in my hand. So if I leave the room kind of scaredly, I'm just looking for my lockdown comfort blanket, and it's going to be all fine. I want to talk about how working memory is stored in the human brain. And this is fundamentally motivated by the idea of information overload. So as we live, we are kind of subject to this constant onslaught of input. And that's a problem for attention, for decision making, but also for working memory, because how do we retain all of this? And the short answer is obviously we don't, because working memory is severely limited, and we kind of, the things that we memorize even for a few seconds barely scratches the tremendous input that we receive. But still, it's worth realizing that there is a huge amount of different contents that we can memorize if we need to, from different modalities, but also across different forms of thinking. Yeah, so conceptualization, sematization, all of these products of a thought can be memorized. And if you want to find out how that is stored in the human brain, you might start with Fuster. This is a kind of old drawing of prototypical neurons that Fuster kind of found in 1973. And actually, we're going to start up with the ones that don't have memory. Yeah? So these are basically sensory neurons. This one is just transiently activated when the cue onsets. Right? So this would be some kind of memory stimulus. And also when it's offsets, and the same kind of for some recall stimulus. You know, is this the same as this? Yeah? Sorry for everybody out there who cannot see my laser pointer. That's just how this works, sadly. This is a more prototypical sensory neuron that basically fires when the thing's on. Same later. Now, what's interesting for working memory are cells that show persistent activity. And this is the prototypical persistent activity cell that basically just prolongs the firing during the delay if it is being memorized, the content that's on the screen, for example. And it only stops when memorization is done. And this is just a different kind of tuning. So here, this cell actually has suppression during perception and activation during delay. And this is mainly here to give you kind of the thought that persistent activity comes in many shapes and forms. There are so many ways neurons can persist over the delay, and some of them look rather weird, and this is just one weird example. Now, what's really interesting if you want to study working memory is actually selective persistent activity. So activity that is selectively representing what is currently being memorized. This could be, for example, this single neuron, again, worked by Fuster ages ago, and that's really interested in green, no, in yellow. And so it fires when you see yellow the most, and also when you memorize yellow, it fires more than in any other kind of situation. And that's what we call by selective. It's, you know, for one stimulus, not for the other. And the same can be done actually across large populations of neurons. So this is a ton of cells actually in the frontal eye fields, and they really care about spatial location. And basically, the red line here is the spatial location that that population of neurons is interested in, and the black one is the location they're not interested in. And you see this differential in the firing, and that's kind of selective persistent activity. Now, if we now take a step back again to our sensory neurons, we know that they come in beautiful patterns. So what you see here is actually V1 and V2, and these are orientation columns. So kind of like pattern responses. So for example, like the green stuff responds to kind of what is that 45 degrees maybe, and red stuff 180 degrees, and they kind of form these pinwheel-like structures. But actually, other beautiful weird patterns can be found in auditory cortex. This is actually tonotopy, so selectivity for different pitches. And the reason why I'm telling you this is that selective information operates on different scales, right? We have single cells, we have population of cells, we have these kind of images done with optical images, right? So this is still in the sub-millimeter domain, but still these are millions of neurons already. But when we are moving towards fMRI, we kind of end up on more an area by area scale, right? So this is the famous Feynman-Paesen picture of lots of visual areas, and what you already see here is that there is a hierarchical structure from early visual cortex, right? No auditory cortex on this picture, I think. It's too many areas actually. And it goes all the way to different visual areas, parietal areas, frontal areas, but also hippocampal areas. So that kind of wide complexity of representation kind of builds a hierarchical grid. And when I mentioned earlier that we would be interested in semantic representations or abstract representations, representations beyond the individual kind of percept that you see. And we're thinking actually in perception, we're thinking about that hierarchy. How an input is turned into something that means something. Yeah? And this kind of combination of a hierarchical structure and persistent activity in many areas has led, again, Fuster, I'm kind of a Fuster fanboy today apparently, to, in 1997, make this prediction of how working memory works. Yeah? I'm calling this distributed working memory because that's what I'm calling it. He calls it kind of network memory, but basically what this says is that there is an entirety of the cortex that's involved in working memory storage. Right? So when you start down here, you have all the primary sensory areas, you have kind of more mid-level areas, conceptual areas, and all of these, in his view, are capable of storing information. Actually, he has this little knack here that he actually turns the whole thing around and says, like, actually, these conceptual representations then can all the way back to motor cortex, so here, and all of these areas have a different form of memory that's maybe more perspective, basically motor plans in motor cortex, or more conceptual plans. All of these can be forms of memory. And the way he kind of puts that idea forward is something I really like, and I'm paraphrasing it here. He says, we are not looking for a memory system, not a thing in the brain that is like a RAM chip, but we're looking for the memory of systems. So all of these systems have a lot of other stuff to do, like detection and attention and all of that, but they also store stuff. And I think he proved to be right. So this is from a recent review where we looked into monkey electrophysiology studies, so the single cell work that we talked about earlier, and data from human imaging showing distributed persistent activity that is selective. Yeah? And basically, the short story is here, it's a lot of places. Yeah? And that already kind of gives credence to this idea. There's a lot of areas could serve as source. And the additional thing that's in Fuster's idea is that there is low-level storage and high-level storage. And it's kind of in that data, where you see lots of low-level features here in early visual cortex, and things that more semantic and verbal, maybe the more anterior it goes. But actually, that's not quite the way it looks in its entirety, because orientations that are given early visual cortex are actually in a lot of places, for example, right? Parietal cortex, frontal eye fields, lateral prefrontal cortex. So there seems to be a way of thinking about this where any given stimulus might have multiple forms of representing them. They could be low-level sensory, they could be spatial, they could be verbal. And maybe we are capable of concurrently holding multiple representations at the same time. Yeah?
Can we ask questions on the way?
Yes, definitely. Yes, please. That's my typical thing is to say what I want to say is actually less relevant than what you want to know.
I have a question that so everyone is welcome. This question. I just wanted to clarify. When you're showing that there's orientation selectivity in several pieces of cortex, does that mean that they're all active at the same time, or is this kind of dependent on other things?
That is a very tricky question, and the answer is I don't actually know. Now, it's sensible to assume that, right? But it could be that, like, because we're always averaging across trials, that some trials in one place, some trials in another place. I don't find that likely. Technically, I only know of one study, one electrophysiology study that actually kind of used the correlation between the two areas and decode on that. And that kind of would say that both of these areas should have information at the same time. But even there, I wouldn't be grounded. It's a reasonable assumption, but methodologically, because in basically none of these methodologies, even with single cells, persistent activity tends to be weak. You don't necessarily see it in every trial in a robust way, so it's hard to argue for that. But you know, the alternative seems weird. So another thing that's weird is being in this psychology seminar and kind of hearing all of this brain stuff. So question is, why does it actually matter to somebody who might not even care about the brain, right? Human behavior is relevant on its own in its own regard. But what I'd like you to take home today is the fact that how exactly this is distributed actually allows for predictions about behavior. Yeah? Because if we have what we call the gradient of abstraction from sensory detail to categorical semantic, there could be a verbal, like the labels here are still kind of putative. But if that gradient of abstraction kind of exists, and we can have representations in a lot of places along this gradient, then the behavior that we output from this these kind of stores actually depends on what of these stores are utilized at any given moment in time. Or put more simply, if you store it kind of heavily in sensory areas, you would assume a more detailed response. If you think it's more in categorical areas, maybe the representation is more adapt to kind of generalize across different similar stimuli, because it's combines these similar stimulus into a category. Right? And I hope that I can convince you of this. But before I do that, I have to give honors to my status as my methods person. That's actually very, very scary to say that. But I quickly want to show you how, in principle, we decode stimuli. This is basically very, very old data, just because it's a simple study to show you how we do it in principle. And then we have this current line of research, which is a lot about what is the nature of the signals that we can read out. So can we tell more than the yellow is different from the green in that neuron, or in that voxel, or in that brain area? So we're going to look at shapes and color for this. And then finally, what can we learn about the mechanism underlying working memory? We're going to look into prioritization / attention for that. And I'm going to make a very putative step into load and other dangerous domains. Finally, if we actually still have time, we're going to talk about memory control, but this is just because I realized that when I tried out this talk, I had 10 minutes left, so I'm like, oh, well, we can also explore those slides. Okay, so how do we decode working memory? For this, I have to introduce you to these guys. This is not an EEG study, this is my study. But these are the stimuli that we're using in the studies. They're kind of complex, multi-characterized fields. Yeah, we like fancy names. And basically, every subject got a set of 4 of them to memorize in our study. And the task was first to memorize them, so you would see 2 of these patterns. Then we have this retro cue. So 1 meaning memorize this one, you can forget about that one. Memorize the first one, forget about the second one. And that is here so that we have control for perception, because both of these are perceived, and as you probably know, fMRI has a very sluggish response, so it reaches into the delay. So what we can actually do is see whether there's more information about this one than that one, and see whether there's actually a specific memory response here. Finally, at the end of that delay, we have this very tricky similarity task. And this is a simplified example. So your goal is here, if you memorize this, to tell me which of these 2 is more similar, and I made it easy for you. This one is the target. And the goal here is that subjects have a heavily detailed representation in their brain. So we kind of push them away from verbalization towards memorize as much of this display as you can, so that you can kind of identify that this little red thing is actually a different location than this little red thing, right? So that's fundamentally the difference between these 2. And actually in the scanner they had much harder trials. It's just hard to do it like on the screen right now. So now is the one method slide. There is another one in about 25 minutes, but this is one method slide. Basically, if you stretch out our encoding period with this retro cue and our task, you see that there is a huge delay between this because fMRI is slow, and we assume there to be some kind of memory signal. And we record fMRI data, and then we look at that fMRI data 1 time at a time. And we're doing this using a search-type approach. So this is us kind of having a systematic way of looking at every location in the brain in an unbiased fashion, by basically starting, at some point, say for example here, taking the pen around it, moving 1 step further, looking at the pen around it, and so on and so forth. So, for example, this pattern could look like this for stimulus A, and it could look like this for stimulus B. And the question here is that actually just random noise, or is in that area, is there a meaningful difference between these 2 patterns of brain activity? And for that we use magic. It's called machine learning. It's also called statistics, because machine learning is just a fancy name for statistics. But basically what we're doing here is if we're looking at our little pattern here, it consists roughly about, I think, 255 voxels or something like this. And we're kind of projecting the activity of this single image here into a 255 dimensional space, which you see here, not because we don't think like that. So I only show you 2 voxels. And in this 2 voxel space, just an example, we plot our different trials for stimulus A and stimulus B, and then our magical classifier draws a separation line between those in a somewhat optimal way. That's basically all the magic that in support vector machines that is doing it somewhat optimally. And you can have a huge debate about what method you want to use. Not that this much. But what we're doing is we have basically a model of what's A and B, which is our separating hyperplane. And then we take independent data and ask, well, is this actually a stimulus A? Because then we were correct. Is this stimulus B? Well, then we would be correct too. Great. If that is true, we have perfect accuracy. But obviously, we're doing this with a lot of different trials, with a lot of different subjects. So what we get out of this is an accuracy of, say, optimistically 65%, realistically 52%, right? But anything above chance is great. So we then take that number, put it here in our image, move to the next step, and repeat until we have a full image of accuracy of information. Yeah? So significant information above chance is there is something in that area that encodes the stimulus. And then we repeat this for all stimuli and for all time points, and we get basically an overall image of where in the brain is information about what's being memorized during the delay period. And that's what we see now. Okay. So at the time, we were totally sold on the idea of the little RAM chip. So it's 3 years ago, sorry. So we thought it would be a prefrontal cortex. It's not. It's actually very much in sensory and parietal areas, right? So apparently our little manipulation in hindsight kind of worked to push us into some kind of sensory representation, maybe some kind of spatial representation. That's kind of the ad hoc interpretation of this. And the time course also looks basically as you would expect it. You don't see much information in the beginning because fMRI is slow, but then later on you see pretty robust signal throughout the day. And we've been doing this a lot. That's basically how we ended up with this giant picture that I probably should have shown here again, with lots of places carrying information about working memory. But I'd rather actually talk about invariants. Because this is now kind of leading towards what can we tell about these signals. Right? And so our initial study, basically, our resonant topic field was kind of the law for the subject. So what is on the top left has to be on the top left, little red dot here, blue there. That precision was relevant. That's actually a relatively weird task, to be honest. So what we actually went for is now shapes. And these are equally obscure and hard to verbalize, but still shapy shapes. 10-sided, randomly generated. Actually, the statistics behind this is way harder than the machine learning to make these in a kind of accurate way. But so hooray for experimental psychology. No, let's not try. No, basically, the task here is, in the beginning at least, it's the same. You see two of these items, then there is this cue with a star-shaped mask around it. This is just to suppress kind of perceptual activity a little bit. And at the end of the day, we have, again, a similarity discrimination task, but it's an invariant task. So subjects have to realize that this, if you rotate it correctly, or it kind of looks like this, so these are more similar. And again, we had kind of a set of stimuli, so you can look at them, how ugly they are, given how hard it was to make them. But this kind of setup with randomly rotated items, and obviously we also randomly rotated the samples at the beginning of the trial, leads to a situation where you can actually do an analysis that looks for invariant representations. So representations of these shapes that are somewhat similar or even the same for different rotations of the same shape. Yes?
Why do you use similar targets and not the same? Isn't it harder to remember only a similar target than?
So the idea behind similar targets is that a identical target has all features the same. Here, actually, only some of the features are the same, meaning that you have to memorize more than one feature, right? That was particularly relevant for the color patterns, where you could basically just look at one spot and see whether it's red or blue. So, yeah.
But there was also orientation in it. The color patterns, there was also orientation in it.
There was to some extent, yes. The kind of also the color patterns have some shapiness inside it, and they have kind of edges that form kind of at least implicitly, right? Because of the way we actually perceive color, which we will talk about later. Even though there's a gradual flow from red to blue in those images, we think of them being edges because we see the edges between color categories as something that exists. Right? So, yes, so there is orientation here for the color patterns, but there we never rotated the items. We have actually another rotation study with the color patterns. If you want to know more about that, I can tell you later. It's not in the talk. Yeah, so the idea between the similarity task is to focus on details. You need to memorize more than one feature of the stimulus. Okay, so how do we get an invariant memory signal? And we do that by actually, these are a set of rotations for this item. We train on a subset of them and test on the rest. Yeah? So we're basically saying is the distinction between these two items captured also by that distinction? And a resonant topic code, right? A very down to the pixels kind of neural code, receptive field in a non-rotated manner, would not be able to do this. Instead, what we would find is an invariant neural code. These are the results. And basically, what we find is, first, that actually very early visual cortex, and it's hard to see here because I'm showing this from the side, is actually spared. So apparently early visual cortex can't quite do this. But LOC, posterior parietal cortex, or what you also call IPS, and the quantum I fields all harbor such representations. And to that, there was a little bit befuddling, not about the LOC, because that's basically the area that we expected, but posterior parietal cortex and the frontal eye fields are, like early visual cortex, retinotopic field maps. So they basically, if you have something up here, my PPC neuron will say, oh, there's something up here, right? Now, such a code doesn't work for invariants, right? So what we kind of guessed was, apparently, they kind of reutilize a field map to maybe represent a prototypical representation of an object or shape. Just a guess. But to kind of look further into this, we did the whole experiment again. But we had a little change because we actually removed the invariant from the task requirements. So the samples would still be randomly rotated, but the target would only be the one that's in the same orientation. We actually had catch trials that checked for whether subjects really memorized the rotation of the item. And then we said, okay, well, something has to change now because it's a completely different task, but it's not. So even in this task where rotation is super relevant, you have basically exactly the same pattern of results. By the way, yeah, no replication crisis. So these signals are also invariant representations, even though rotation matters. The only difference between these two experiments was actually that there was this tiny blotch in the peculius. This is now the medial wall, it's kind of like behind here. Actually has information about the rotation, and that's actually not particularly reliable, right? So I actually didn't put this in the paper initially because it's technically not something that our reviewers made us put in the paper, so I'm putting it on the slide, and I think it's actually there. Because it's actually significantly higher than what's not here. So probably it's there, no guarantees. But if that is true, that would imply that in this task where there's two properties that are actually relevant for the task, the shape itself and its rotation, you are actually disentangling these two representations. Rather than just having a kind of retinotopic code of the shape as you saw, you are actually having maybe a prototypical representation of a prototypical rotation of the shape, and then have a separate representation of the rotation, which is weird. But apparently it's efficient, because otherwise why are our subjects doing it? And what this implies is that the representation of an item actually can travel up and down the gradient of abstraction, utilizing different areas for different purposes, depending on the task demands. That's how we interpret this kind of task. And, you know, the problem is that we have this example, and we're looking for more. It's hard to get to these examples, because FMI takes time, basically. So we don't actually fully understand how that works, but that's basically one of the biggest goals that we're after, is like understanding how contents travel around the brain in what I kind of over here tend to call like mental Tetris. I can put this there, this there, this there. Okay. Now, I also promised you that I would talk about color, and here we kind of more directly wanted to test one of our assumptions about the gradient abstraction, basically that there is a gradient abstraction. There are different representations, different brain areas representing the same content of different kinds. And for that, we went for color, mainly because color might be the most used stimulus in working memory research. Particularly in behavioral research, it's just a bunch of studies. I'm not going to go into the details here. It's more about realizing that how ubiquitous color is used as a stand-in for visual working memory. I'm trying to convince you that visual working memory isn't quite visual, because it in part is categorical. And as always, when somebody is doing some cool FMI work, it's cool, somebody else has done some cool behavioral work before that. And this is basically my favorite paper on color ever by U of A. And the thing that Gil argued for is that there are two forms of color memory. One sensory and one categorical. And the way he argued for the categorical part was by realizing that if you have kind of lots of colors on the color wheel, and you don't kind of, or you ask subjects to recall the colors, so basically to click on the color wheel to tell, after the delay, to tell the experimenter which color they remembered, there are these biases right? So for example, this kind of greenish yellow is actually reported more green than it originally was, consistently across trials. And Guido said, believed that that was kind of the result of an attraction by a category, right? It's kind of in the middle, it's probably green. And that kind of phenomenon, he tried to model with completely independent data. So he used information about color categories. I will tell you in a second what actually he was using, because we're using the same, to form a behavioral model to predict these biases. And as you can see, it actually quite successfully did so. And because we liked the study a lot and we were lazy, we actually did the same study and put it in the same. Well, this was me being lazy, not Chang, because this is work by Chang Yan, who is now Dr. Chang Yan, and actually I think she defended either here or over there. Very proud of her and this work. And in the experiment, it kind of looked the same as before. So if you're getting bored by this experimental size, you can kind of pass out, that's fine. But basically you have two samples, one random queue, and the big change is not a similarity task anymore. But it's basically what I said before, you have a color wheel. If this is what you're memorizing, you kind of point out at this part, color wheel, you check, and you know, okay, that's what you've been learning. Now, we also use behavioral tasks outside of the scanner. These were stolen from the viewer's work. One was category naming. So you would see a color, and then you would have to select one of these color names. And this would be the distribution of color category naming. So basically, all of these colors are mostly green. These are mostly blue, and then there are these intersection points, which are kind of the boundaries between categories, where you already see that categorization is, we think of it as a kind of like a boxcar kind of way. It's either one or the other, but in practice, there's uncertainty that comes with categorization, and that's kind of reflected by these overlaps. And these are the boundaries across subjects. Y-axis the different subjects, and then on the right. This is category identification. So I would prompt you with a color name like pink, and you would tell me what pink is. Yeah? Again, this is basically the same distribution. Other than that, this is kind of focusing on the most green, most blue, most purple, most pink. Basically capturing what is the most intense form of that color. Again, for that particular color, basically the peak of that representation. You see some variants, but overall there's kind of the same picture of the subject. So why did we do that? What we wanted to do is we wanted to make an encoding model. Now we're getting to the second method slide of today. I'm also scared of it. We can be scared together. That's okay. So basically what we did is we just averaged these two distributions and to make a prediction of how we believed color categorization would work in the brain. So, kind of to put it kind of simply, is we would say that okay, there's probably some kind of green neurons. These green neurons fire a lot when it's very green, and they fire less and less the less green it is. Basically, just taking this data into account, kind of taking this peak and that distribution together. And given that, we have the simplified idea of color representation that, well, there's green neurons, there's blue neurons, there's purple neurons, and then we would say for probably an fMRI voxel that would consist of a mixture of those. So we could explain the variance in the fMRI signal by having in every voxel a mixture of these. And this is I'm gonna show you how I'm doing this. First, I'm quickly telling you, we tested the categorical model against sensory models. So we were looking at two forms of color memory. So this is just a very standard way of conceptualizing sensory representation. I'm not necessarily saying that it's a good way, because what actually do we mean by sensory? Different debate. But a lot of people have been using this, so we took this. So if you, for example, have activity from B1, you have a lot of trials with a lot of different colors and a lot of different voxels. And we would basically, yes. Oh, I'm sorry. Yes, please.
Sorry. I don't understand how similarity was defined validated. How do you know that the chosen shape was more similar?
Okay, so we're jumping back 15 slides. So, sorry, whoever asked that question for not realizing the question earlier. Yeah. So, no, I'm already here. So the thing is, we just told subject what's similar. That's kind of the simplistic answer, because obviously subjective similarity can be very hard. We actually had subjects like kind of 2 or 3 subjects that couldn't do any of these tasks at all because they had different conceptions of similarity, I think, or maybe they just didn't want to do the task. I'm not sure. It was a really hard task. But basically, we trained them hard outside of the scanner with feedback. We didn't have feedback in the scanner, but outside of the scanner we had feedback telling them, no, you got it wrong, you got it wrong. And that's how we define similarity in the concept of this task. We did this with very simple correlations. I think it's just simple correlations because basically what these things consist of is 10 values, which is the distance of the center to the individual corners. So basically, every one of these shapes are defined by an array, by a vector like this, and you can just correlate the similarity for 2 shapes. Now, for the invariant task, it would be kind of correlating all possible rotations, yeah? And for the rotation-specific task, it would be the in-frame kind of correlation, so just without any possible, right? Just correlating as you see it on the screen right now. Does that answer the question? Yes. Okay. Okay. Yes.
I'm up with the colors because I know that we see colors differently depending on what context they're in. Maybe with the blue frame, you can see people in the gray frame or whatever. Did you use this brown gray in the original study, or how did you sort of
So after spending roughly 8 hours in the scanner calibrating, I was happy that it wasn't blue-gray anymore, because I only then realized that I've been scanning with a blue screen for 5 years. Because the screen that we're using in the scanner is actually very blue. It's just you don't realize because you have no other context, as you say. So what is being presented there as gray is gray. It's also actually the reason why the colors are that light, because when you then shift the color space, there isn't that much space to make them darker, kind of. Yeah. And that kind of is what we did here. We didn't use any brown, gray. Actually, that's the first time that I've heard about it, because probably I'm not as good as color as that. No, yeah, it's well, like it was supposed to be medium gray. Like the thing about what we showed in the scanner and what you should see here is all of these colors are not actually the way you should saw them in the scanner, because we only ported the RGB values from that very weird screen to here. So yeah, it's hard. But yeah, we gave it our best, basically. It's still kind of puzzling, weird, and it's just, and actually one of the colors that we're using is slightly misaligned. If the paper comes out, you can see like, that's the one that kind of at 4 o'clock in the night I said like, fuck it, give me that. Okay, so back. By the way, thank you for kind of giving me some respite from the evil method slide. Okay, so we're going to do this easy. You have data from voxels and colors, and we just do a very simple general linear modeling. So we basically what we're getting is parameter estimates, right? So what this says is this voxel here, voxel number 1, cares most about the color channel. Cares little about color channel 1, cares a lot about color channel 2, cares little about color channel 6. So in other words, if color channel 2 is orange, it should respond most to orange. And basically this is just toy data, so I probably fucked it up. So the idea is that this describes kind of the mixture of different neuron types in our prototypical simplified categorization model, and how the voxels weigh on this. Now, what you could do now is actually turn the whole model around, invert the encoding model. Now, I'm not sure this is probably interested to one person in the room, hopefully one. What we would then do is kind of predict the colors. So we take independent data, take these weights, and kind of project it back and say like, okay, in this trial we had red, in this trial we had blue. We didn't do that because what we're interested in is actually not predicting the outcome or the input of the system, but we are interested in the internal state. Like in between the input and the output, there are multiple internal states which neither reflect the input or the output perfectly. So actually doing that would kind of destroy the purpose of the whole thing. Instead, what we're doing is actually basically just cross-validating these parameter estimates. Basically saying across different runs, is this pattern, still a pattern analysis is magic, consistent across different repetitions of the same set of colors. And shout out to Carsten Allefeld, who developed this method, which is called CD Maneuver. He has a toolbox, use it because it's great. You can do a lot of fancy shit with it, relatively easily actually. And the base idea is that we get out a pattern distinctness. So is the pattern distinct between color channel 1 and color channel 2? Because in an environment of pure noise, when the brain area in question doesn't care about color at all, we wouldn't actually see any pattern distinctness, right? Because it's just random noise, it's just kind of all the same, right? So if these patterns are distinct between the color channels, we have a robust color representation. And the cool thing about pattern distinctness is that it kind of also serves as a measure of explained variance. So basically, how much variance does our encoding model explain of the fMRI activity? The short answer is very little, because fMRI is a very bad signal to study. But when it comes in a relative measure, then it becomes interesting. So let's look at some results. But we looked only at V1 before V1. And in V1, you see that both of these encoding models, our sensory model and our categorical model, are kind of forming the same. And this could kind of then lead us to say, yeah, categorization in V1, cool paper title. Problem is, these two models are highly correlated, because they're basically coding for the same thing, just in slightly different forms. So as long as there's a significant difference between the two, we don't know which of these is actually better, right? But thank God, there's a star. A star was born. We have a significant difference between categorical models and sensory models in V4 and V1. Meaning that already in the relatively early sensory areas, we see that during working memory, representations are more categorical than sensory. So basically that applying some kind of gradient between this area where we don't quite know and these areas. And the really striking thing was that we also did the same thing in underlay tasks. So you would see the color and report it straight away. And in this task, we actually can see the pattern. We almost see kind of the opposite pattern, such that we actually in V1 we find a significant interaction effect, meaning that the representation in working memory in V1 becomes more categorical with memory. Or if you kind of slightly want to overinterpret it, memory makes that area more categorical. And that was quite exciting because, okay, well, first of all, the gradient abstraction thing kind of worked. We can find categorical signals, and they seem to be more robust than our sensory signals. And apparently, also what memory does is actually it pushes us to more categorical representations within these areas. How exactly? Another kind of hard to get to question. We're working on it, hopefully. And talking about working on it, because the other most used stimulus in working memory research are gratings. Poor Joanna here is now kind of letting subjects name gratings. And like I'm really suggesting whenever she gives a talk here to come, because she has a huge kind of list of weird responses to what you can call gratings. This is just basically the big ones, yeah. So it's kind of what you think, like straight and horizontal and left and right and stuff like that. We kind of want to see whether we can do the same thing with gratings and spatial locations, and very much looking forward to what Joanna comes up with, because that's even more weird than the color. All right. Yes, yeah, of course.
Briefly about the color thing is, I'm not sure if I understood the categorical and sensory explained variance that you showed was compared with the distributions of people, the two distributions that you showed of where is the green and where is the maximum.
Yeah, well, these kind of, this is the prototypical color, that's the boundary color, that's just a thing that I showed to explain it. We actually used the full distributions of these two tasks, the category name and the category identification task, to inform the full encoding model.
Okay, good. That was in the front. The multiple phase.
Well,
what is then here the difference between sensory and categorical?
Okay, so I actually probably didn't show the sensory model long enough. This is the sensory model, this is the categorical model. So this is kind of, the sensory model basically assumes that color is a continuous space, there are no kind of boundaries in there. Yeah? It's a uniform space, and this kind of suggests that it's not a uniform space. What it actually doesn't suggest that is that our silly model is the best model for color categorization, because that seems ridiculous. We only have like 6 color terms in there, and like humans are weird with language basically, and so it's probably more complicated than this, but at least it works better than the other thing. So great. Martin, can you quickly tell me the time?
It's 4.
So we have another 15 minutes. Yeah, because we planned the question. So that's great, because I was kind of shocked because this thing has been running for an hour and I was like, oh, I have to be done soon. Brilliant. So now we get to the third part. And the question here is basically how we kind of now have learned more about what kind of representations are we looking at. Apparently the whole more detailed sensory areas, more categorical further on, hopefully kind of works. Thank God there's other people also working on this. We also have very nice data. Our idea was now to study what changes when areas are utilized. That was our fundamental motivation, to find out kind of what happens when memories shift around, and why do they shift around. And our first kind of goal for this was prioritization. But actually, for the study, there was another goal, and I want to be very open about this, because at the time, and actually still today, sadly, there was an ongoing debate about null findings for so-called understanding memory items. So these are 2 studies, one by Joshua LaRocque and one by Michael Wolff. And both of these studies kind of had a situation where you have 2 items of memory, but one is relevant, the other one isn't as relevant, but you still have to keep in mind. And what they found, or actually not found, is that they couldn't quite decode the unattended memory item, the less prioritized item, right? So this is kind of a weird plot, but you have to trust me that this is kind of the point of interest, and the fact that this blue thing and this gray thing overlap mean that there's, in this data, there's no information about the UMI, but there's plenty of information about the AMI, the attended memory item. And this is kind of the same here in EG from the Stokes group. And that happened like 3-4 times in a row. And people had this idea about it. Because when I talked about cluster and persistent activity, that's basically the standard model of how working memory works, right? Basically, you have a bunch of cells and they fire, and they're kind of like it's a neural circuit jerk. You're like, here, you have to fire, we all fire together, and then we are all happy, and we retain information. Now there's an alternative idea, which actually also Fuster introduced at some point with is the idea that there is activity silent working memory. And that is that these neurons actually don't fire, but they retain the information that they had fired recently in this synapses to each other. Yeah? So it's basically like putting a light shoulder, like a hand on the shoulder, saying like, yeah, I'm going to be back soon. And in theory, this could happen without any firing happening. And that's why it's called activity silent work memory. And so what the poster group and the Stokes group kind of argued for is that, well, we didn't find the unattended memory items, probably it's activity silent. And we talked about magic, that actually feels magical. And also we had some doubts about it, so that's why we wanted to do that ourselves. Also, we had this prediction that actually is something completely different going on. I'm going to see what. First of all, this is work by Paulina. Paulina is a really great former master student who kind of pulled this project through. If you want to be a great master student, don't be like Paulina because she worked hard on this. So in every given trial, you had 2 orientations. We're finally at orientations. And we masked them a lot because, again, we wanted to suppress the kind of visual after images and stuff like that. But then actually there was an initial phase where you have to memorize both of them. Then there was a queue, and And the red side here is the one that you should attend to. So this item up here is suddenly the relevant item, because shortly after there would be a task and you would have to tell kind of, is this thing rotated clockwise or counterclockwise relative to the one that you saw earlier. Now, the problem was, there was another cube later on that could either be the same or the opposite, such that you actually had to keep the other thing in mind. So in this critical period, you have two things in mind. One is attended, and the other one is less prioritized because unattended is probably not a thing. Okay, and we basically used the subset of areas because orientations have been studied over and over and over again. We used the subset of areas that we kind of knew cared about orientation. And looking at V1 to V4, we could see that, great, we have silent working memory. Yeah, so the alternate item is here on the left, super clear, super strong. The UMI is nothing there, at least in this analysis. And so we could just go home and kind of announce to the world, statistics prove that null findings tell us that there is silent working memory. Unless we had this silly figure still in our head, because the silly figure told us, okay, there's a lot of other places where you can also. If you look at other places, particularly the IPS and the frontal I fields, you actually see very robust information about unattended memory item. Actually so robust that these are not significantly different from the attended memory item. And we think actually that this pattern of results, I promised you to come back to the point where saying like, okay, the utilization of neural areas is relevant for behavior. We find it there. It's just one slide, right? This is the one. In the repeat trials, performance was kind of skyrocketingly high, better than the switch trials. And that comes with some other caveats, but the one interpretation that you can have for this is that the representation in V1, V4, you know, or the detailed area, the thing that has high precision over representing contents, actually served that item better, such that it is recalled better later. And we also have to find out more about this because there is a silly caveat here. But I think that that's what's happening here. Okay, and we're slowly coming towards the end because I just kind of wanted to show you two more slides over another paper that we're after. But before, yes, prioritization kind of gives a focus to sensory detailed representation. So those last two slides are about load and stimulus modality, because in the end, what we are always after when we're studying working memory is the question, why are we so bad at it, right? Like, I think the nicest comparison that you can do is, like, who here remembers the Tamagotchi? Who knows what a Tamagotchi is? Yes, I'm nodding. The Tamagotchi has a severely better working memory than you have. Like, it's a couple of K, it's actually horrible. But given what you can do in working memory, it's really good. Yeah? So why is it bad? And I'm not going to answer that now because I don't know why. But one step to doing that is actually looking what stimulus composition does with neural representation. In this slide, here actually at some point made to write the grant that I'm currently on, because these are actually two completely different experiments. On the left side, we have some sort of unimodal task, meaning you have two orientations. In this task, you have an orientation and a pitch that you're memorizing. And what struck me here was that here we have this really heavy involvement of interior areas, IPS and frontal I fields. So this is orientation, right? Yeah. Same content, but totally different utilization. Because here IPS and frontal I fields do little. Yeah? I'm not saying they're doing nothing because I don't interpret null findings the same way my colleagues do. And basically, if that holds true, and right, like that's kind of like a hamstrung comparison. Think of it as pilot data, basically. But if that holds true, then it means that unimodal load leads to a utilization of more interior areas. And when we have looked into that example, right, prioritization makes it better by pushing it down. Then unimodal load pushes it up, leads to worse behavior, right? And this is kind of what Chapurian is working on in the lab. And this is just kind of our first data set that we're having here, and apparently that part holds true. Again, like we are bad behavioral psychologists, so there's a bunch of caveats that come with this. We're working on more iterations to find out kind of whether it actually holds true. But before that, we have to go to the scanner and actually scan the study because it's a thing we're also excited about. And that leaves me at the penultimate end of this talk. And now, click the button here. Oh, here we go. Leaves me to thank my collaborators, particularly John Dylan Haynes, who kind of carried me through my years as a PhD and postdoc.